At Forage Genetics, we utilize a Pedigreed breeding system. We perform phenotypic and genotypic recurrent selection along with marker-assisted selection. In a Pedigreed breeding program, what we're doing is keeping track of the parent plants and the progeny or the families those parents produce. This is a 2-year-old nursery. We typically leave them in for 4 years. Over that 4-year period, we're monitoring each family and each plant for pest resistance, for winter hardiness, for disease resistance, and at the end of that 4-year cycle, we'll select 1 to 2% of the best plants, and we'll take those into the greenhouse to make pre-breeder seed. With pre-breeder seed, we're targeting 10 grams of seed, and we can plant that seed in a targeted disease screen to further improve disease resistance. So we'll subject those seedlings to Aphanomyces or anthracnose. Could be an abiotic stress such as salt, and what we're trying to do is find the genetically superior plants or seedlings in that situation. Those plants or seedlings will serve as parents for our breeder seed that we'll produce in Idaho. When we make that breeder seed, we're targeting 5 pounds of seed, and that will serve as a planting seed that will enter into our multi-state testing location program. We plant them into forage yield trials. We'll cut and weigh them for a period of 4 years. And at the end of that 4-year period, we can look at our forage yield and our forage quality, and we'll advance 5% of the best performing experimentals, and those will become a commercial variety.